Good morning. I'd like to first thank the abstract reviewers who played a major role in helping us to evaluate all of the submitted abstracts and help us select the top oral presentations and poster presentations for the poster tour. And I'm very happy to be able to be one of those to share with you my work from Ottawa today. So I'm going to introduce another component to this T-cell story, and that is looking at the condition of the liver and how it could affect T-cell responses.
You like the right arrow.
Okay, so we do know that chronic liver disease has widespread effects on the immune system, and this slide is not meant to scare you about that sort of interaction, but we know that cells in the two major arms of our immune system, that being the innate immune arm and the adaptive immune arm, we can find dysfunction in nearly all of these cells in the things that they're meant to do, the things that they're meant to secrete, and the interactions that they have with other cells of the immune system. And we can observe this both in the liver, in our lymphoid organs, and in circulation. And so my research is looking at evaluating specifically the CD8 T cells in this relationship. And so we do know that there are a number of complex clinical outcomes that are associated with advanced liver disease, with the increased scarring of the liver. And so many of these can be covered today and throughout the weekend if you're staying for the CLM. And that CD8 T cells can play roles in these conditions through the mechanisms of their action, which include their ability to kill their targets, whether they're virally infected cells or cancer cells. And they do this through the production of lytic molecules like perforin or granzymes, or their engagement of death receptors like fast-fast ligand or TNF receptors. They can also be important players in the modulation of immune response through their production of important cytokines, including interferon gamma. And so we can find a role of T cells with other immune cells in many of these clinical outcomes. So there is incentive here to understand further how CD8 T cells can be affected by chronic hep C and advanced liver disease and the outcomes that follow. And so it's very well described that virally specific CD8 T cells have significant dysfunction in chronic hepatitis C infection. And so that's been very well documented. And some of this has also been extended to the larger pool of CD8 T cells in circulation and in the liver that are not specific for virus. But the thing is that most of these studies have been done in individuals with minimal liver damage. And so some of the early studies that I did with my clinical collaborator, Curtis Cooper, we've been working together quite successfully and productively for the last few years. This is a beautiful picture. And in the first study that we did with my first graduate student, Stephanie Burke, among other things that we measured, we fortuitously found this relationship that CD8 T cells, not necessarily specific for hepatitis C virus, actually had lower expression of an anti-cell death marker, demonstrating that perhaps there could be an influence of the liver disease severity in a function of non-HCV specific CD8 T cells. So we've decided to look at this even further by a larger study to look at liver condition. And so you need to be reminded just quickly, it's not a lesson in pathology, but how liver fibrosis is scored using a Medivir system. And in hepatitis C patients, this is principally done now through ultrasound-guided elastography to look at the liver thickness. And so essentially you can have a range of liver scarring, with minimal scarring being a score of F1 with very little effect on liver function. And this can range all the way up to F4, where there's substantial scarring termed as cirrhosis. And this can indicate the possibility of progressive loss of vital liver functions eventually. And so our lab is aiming to try to understand how a state of liver fibrosis can contribute to altered immune function, and in order to be able to identify targets that could help Canadians and others who are affected by advanced liver disease and to minimize these clinical outcomes. And so the hypothesis here is to test whether there is generalized dysfunction of CD8 T cells associated with liver severity. And so our more recent work was done to evaluate this with my research associate, Agatha Vrankovic, who is here today, but she won't be wearing her sombrero. Anyhow, she brought together some data from a former master's student as well to put this work together. And what we were able to demonstrate is that if you look at individuals who have cirrhosis, in the red here, Untreated, chronically infected, they produce a significant amount of lytic molecule perforin in a number of these CD8 T cell subsets in vitro, whereas their counterparts with minimal fibrosis produce a much significant lower amount of perforin similar to that of controls. This was in part alongside increased systemic inflammation in these individuals where we saw elevated levels of inflammatory markers like TNF alpha. And so we were also able to identify in this same study that if you looked at these individuals 1 year later after the initiation of their DAA therapy, and all of these individuals were cured, in that the advanced fibrotic group, there seemed to be sort of a retention of this hyperfunctionality, as we've called it, in the high fibrotics, whereas this was not the case in the minimal fibrosis. And this has also been partnered with a sustained inflammatory picture in the plasma cytokines. So this has generated for us a series of questions, whether or not bulk, non-necessarily HCV-specific CD8s are inherently dysfunctional in advanced liver disease, and could a pattern of gene expression identify potential sources of this dysfunction? And would this pattern be reversed after you remove the liver insult, as the data, our functional data would suggest that is not the case? And so here is our hypothesis to test this gene expression pattern between these different patient populations. So we looked at patients, the same group with the few cells that we had left after that long study. We managed to get 3 patients from each group before treatment and 1 year later. So they had minimal fibrosis or advanced fibrosis. We collected their PBMCs, their blood mononuclear cells, isolated their CD8s, cultured them and stimulated them in vitro, isolated the RNA and sent it for sequencing so that we could look at gene expression. And so this is where I'm out of my comfort zone looking at gene expression, and so I'm really trying to digest this data for the last little while. But big picture is that the principal component analysis, looking at the most variable 500 genes between these two patient groups before treatment, identified a very clear and distinct pattern of gene expression across these 500 genes between individuals with advanced fibrosis compared to minimal fibrosis. And so I've been staring at this for a long time, not really knowing what to do. And thankfully, I've gotten some saving by my recent graduate student, Jeff Lee, who's really delved into this data set and generated some fancy pictures. So essentially what we've been able to show is that there are about 88 downregulated gene families in the cirrhotic patients compared to the individuals with minimal fibrosis, and a ton, 356 upregulated genes. So now we can kind of sort of quantify the directionality of these. And so through pretty pictures, we can show that on the left here we have the gene expression patterns, that being the upregulated genes in red, the downregulated genes in blue. And you can see that the patterns of the cirrhotic patients listed on the left are significantly different than that of the minimal fibrosis patients. And so without going into too much detail in the interest of time, what you do with this data is then you take these gene expression patterns and you put them through databases to determine which gene pathways are frequently engaged and which ones are statistically significant, which is the key. And these are as good as the databases that are generated for this. So there are some limitations in preciseness. But what Jeff has been able to show through staring at these data sets is that a number of these signaling pathways that are quite important, in fact, to T cell receptor signaling and such, have been popping up to be significantly upregulated, as shown here in a molecular function analysis. Lots of phospholipase signaling engaged here, and we'll kind of tie this up a little bit later. Another type of analyses, like biological processes, indicating second messenger signaling, migration signaling, interesting engagement of nuclear division as gene expression differences, and the ability to transport RNA and to mobilize your actin cytoskeleton within a cell, which has a lot to do with cell division as well as cellular function. And so the last analysis continued to pull out some cytotoxicity gene expression sets, which satisfies our functional data in vitro, and some more cell cycle and cell metabolism signaling pathways. The last data set analysis that we subjected this set to was something called gene set enrichment analysis, which sort of does more of a ranking of gene expression patterns and looks at statistically significant differences. And so we have a number of potentially interesting genes identified through this type of analysis, focusing on those with the highest statistical significance through p-value expression. So we can see gene expression related to cell survival, cell cycle again, and cellular metabolism and T-cell receptor signaling, as well as upregulated genes. There are a number of these associated with interferon responses. Again, the mobilization of your cytoskeleton at the surface of the cell to release lytic molecules, and the engagement of this hedgehog signaling pathway, which actually has been recently shown to have a role in cytotoxicity. So these are all kind of a lot of gene patterns associated with functions that we were actually seeing in our in vitro culture. So we have a few hits here that we could pursue in mechanistic studies going forward. So to summarize, this includes cell cycle anomalies, actin mobilization, cell metabolism, and other aspects of what we call non-specific T-cell activation, because again, this was the engagement of cells in the absence of antigen. And the last bit, and the data we have for this is very, very minimal, but we did have some individuals who had pre and post samples still available in our biobank. And so we found that in fact these gene patterns, whether you had minimal fibrosis or advanced fibrosis, appeared to be static 1 year later. So they're not reversing their gene expression patterns despite the fact that they've been cleared for their virus for nearly a year. So in conclusion, CD8 T-cell hyperfunction is indeed associated with the severity of your liver disease and chronic hepatitis C expression, and that this is very significantly different in cirrhosis versus minimal fibrosis, and that these patterns may be stable after cure. So the who cares element of this has been nicely laid out through Ellie's talk in emphasizing the importance of T-cells for vaccination. Our information adds to the growing knowledge that there is indeed a lack of immune restoration in chronic infection, whether the virus is controlled, as in HIV infection, or cleared with DAA therapy. And so there are a number of articles that have been published that demonstrate this either at a specific level or more broadly. We could have predicted this through John Weary's work with the chronic LCMV model in mice, where he has been able to show that when you clear this virus, there is definitely a genetic imprinting of these CD8 T cells that make it hard-pressed to have them ever be the same again after that chronic trauma. And so this is going to have consequences, of course, for hepatitis C reinfection, developing chronic disease again, whether or not these individuals will be responsive to these incoming T-cell vaccines, and whether or not it contributes to their risk for liver cancer or their responses to immunotherapy, as well as our mouse models would suggest in the work I did last year or showed to you, is that these T cells will be refractory to immunotherapy. So I'd like to thank the members of my lab and my clinical collaborator, of course, Curtis Cooper, and a huge thanks to CanHepC for their pilot funding, which has led me to be successful with CIHR this past year. Thank you.
Now we open to questions.
So, Angela, it's very nice you describe fibrosis. What about inflammatory infiltrations in these livers? It were visible or completely cured?
So you're asking about what is the scoring of inflammation in these individuals.
Yeah.
And so certainly scores of inflammation don't necessarily go hand in hand with your scarring. At least in hepatitis C, because we don't perform liver biopsies any further, we don't have the ability to, at least as much as I can determine, give an inflammatory score through the ultrasound guided. And the only way that you can really do that is you need to do a biopsy, and that's just not really in our clinics standard of care at all. Certainly in fatty liver disease, there can be joint information now on liver fibrosis and inflammation, and so we'll be looking to tie those relationships together as well.
I understand, okay. So biochemistry, there was any liver biochemical injury with these cases which was different?
Through their liver enzymes.
Through the liver enzyme, yes.
So we haven't. We can get that information and look and see if that. But there sometimes some of these enzymes can be a little bit of a moving target, particularly in the cirrhotics. But we can take a look and see if there's a relationship. Thanks.
Okay.
Hi.
Hi,
So
Dr.
great RNA-seq work. Have you checked or are you going to check about the protein level as well? When you talk about RNA transcript going upregulated or downregulated, often those messages don't translate to the protein level, which
Yeah,
are
for
actually
sure.
the molecules
So that would
actually
be-
do the magic, right?
Yeah, that would definitely be part of the validation process. So certainly these gene expression sequencing analyses don't inherently couple with the protein thereafter. It's something you have to go and investigate. So for sure, part of our validation process will include that. Thank you.
Hi, Angela. Hugo Soudan, St. Justin Hospital. I'm intrigued by your CD8 T cell hyper functionality. In one of your first slides, you showed us increased perforin staining on the CD8 T cells. Did that also show up in your RNA-seq experiments?
Yeah, strangely enough, at least in the RNA sequencing at that particular time point, we didn't see an increase in perforin RNA expression. So maybe it required like longer time to induce. I'm not quite sure how to explain that anomaly, but certainly the other factors that are involved in releasing these markers were altered. So this is something that we're looking at in more detail. We might go through more specific PCR analyses to double check that.
Yeah, and if you look at perforin staining, because what you showed us is frequency of perforin positive cells, is the mean fluorescence intensity also increased?
Yes, I believe the mean fluorescence intensity was also statistically increased.
Thanks.
I'm sorry, we have to move on. Though we can discuss, like later.